There is a point in Earth history where the fossil record begins to feel almost recognizable. And then, if we move only a little farther back, recognition falls away. We leave shells, teeth, claws, articulated limbs, and obvious eyes behind. We leave forests and fish behind. Even the familiar logic of predator and prey becomes uncertain. What remains is a world covered by shallow seas, microbial films, low continents, and soft-bodied organisms, whose shapes seem to belong to a different set of biological rules. This is the Ediacaran Period, the last major interval of the Precambrian. It began about 635 million years ago and ended at roughly 539 million years ago. The dates sound abstract until we compare them with the history that followed. Dinosaurs would not appear for more than 300 million years. Land plants were still far in the future. There were no reefs built by corals, no schools of fish, no forests of seaweed in the modern sense, and no animals walking across land. Yet this was not an empty planet. It was a world in transition, and the transition was more complicated than the old story of a sudden Cambrian beginning once suggested. For much of the 20th century, the Ediacaran Biota were often treated as a mysterious prelude. Strange quilted impressions that appeared, flourished briefly, and then vanished before the Cambrian explosion. That framing was useful because it captured their unfamiliarity, but it also encouraged a misleading idea that the Ediacaran was merely a waiting room for real animal life. New fossils, new chemical analyses, better dating, and more detailed studies of ancient ecosystems have changed that view. Some Ediacaran organisms probably do represent branches of life with no living descendants. Others appear to be genuine animals. Some may sit close to the roots of groups that later became familiar. And some fossils remain uncertain enough that serious researchers still disagree about what kind of organisms made them. The uncertainty is not a weakness in the story. It is the story. The Ediacaran gives us a rare look at evolution before the modern marine world had settled into its basic ecological patterns. Bodies could be built in ways that later became uncommon or disappeared. Seafloors could remain covered by cohesive microbial mats because sediment-churning animals had not yet transformed them everywhere. Large organisms could grow without shells or bones, leaving behind impressions only when unusual conditions preserved them. Communities existed in which rooted, frond-like forms rose from the sediment beside flattened mobile organisms and strange radial bodies that do not map neatly onto modern phyla. The planet itself was also emerging from an extraordinary climatic past. Before the Ediacaran, Earth had experienced severe Cryogenian glaciations, including episodes widely associated with the idea of snowball Earth. Ice reached low latitudes, oceans and atmosphere changed, and the aftermath reshaped global chemical cycles. Exactly how environmental change interacted with the rise of animals remains debated, and simple one-cause explanations are not supported. Oxygen availability, nutrient cycling, ecological interactions, developmental innovations, and changing ocean chemistry all matter. What is clear is that the late Precambrian was not environmentally static. The stage on which early animals evolved was changing beneath them. To enter this world, it helps to remove almost every visual expectation we carry from modern nature documentaries. Do not begin with a giant predator. There may not be one. Do not search immediately for eyes looking back at the camera. Many organisms probably had no eyes. Do not imagine a reef crowded with hard skeletons. Most bodies were soft. Do not expect sand to behave exactly as it does on a modern beach. In many settings, microbial communities stabilized the sediment into matgrounds that could preserve delicate traces and support unfamiliar ecological relationships. Now picture a shallow sea more than 500 million years ago. The water is dim but not black. Fine sediment lies below. A leathery microbial mat spreads across the bottom in patches. Some organisms rise like fronds from holdfasts. Others lie flat against the surface, their bodies divided into repeated units. A small mobile creature crosses the mat and leaves a faint path behind. Nothing in the scene is trying to resemble the future. It is simply alive, on its own terms. That distinction matters because the Ediacaran was not a failed rehearsal for the Cambrian. Evolution does not work toward a predetermined caste. The organisms that lived here were adapted to their own environments, constrained by their own histories, and exposed to ecological pressures that were only beginning to intensify. Some lineages may have persisted across the boundary into the Cambrian. Others disappeared earlier. Others left descendants so transformed that the connection is difficult to recognize. The fossil record is incomplete enough that every confident family tree should be treated with care. In the last few years, the picture has become even more interesting. Fossil assemblages reported from South China have provided unusually detailed windows into the final Ediacaran, close to the transition into the Cambrian. The Tongshan Lagerstätte, described in 2025, includes a diverse terminal Ediacaran community and an uncommon style of preservation that can retain carbonaceous information from soft organisms. In 2026, the Jiangchuan Biota was described as a transitional fauna containing both distinctively Ediacaran forms and organisms with features more reminiscent of Cambrian animal groups. These discoveries do not erase the Cambrian explosion. They complicate its beginning. The question then is no longer simply why strange Ediacaran creatures vanished. A better question is how a world with no familiar animal ecosystem became a world increasingly filled with movement, feeding, burrowing, skeletons, predation, and recognizable body plans. To answer it, we have to begin not at the end of the Ediacaran, but with the conditions that made its strange seafloors possible. The Ediacaran begins in the aftermath of one of the most dramatic climatic chapters in Earth history. During the Cryogenian period, which preceded it, the planet experienced at least two major glacial episodes, commonly called the Sturtian and Marinum glaciations. Geological evidence indicates that ice reached very low latitudes. Exactly how completely the oceans were sealed by ice remains debated, so the popular image of a perfectly frozen white sphere should be treated as a model rather than a photograph. But by any reasonable reconstruction, these were extreme events. When the Marinum glaciation ended around the beginning of the Ediacaran, melting ice did not simply return Earth to a familiar climate. Continental weathering, ocean chemistry, nutrient delivery, carbon cycling, and oxygen conditions all responded. Thick cap carbonates deposited in many regions record a strange post-glacial world. Later in the Ediacaran, additional environmental disturbances occurred, including the Shoreline carbon isotope excursion, one of the largest negative carbon isotope excursions known from the geological record. Researchers continue to debate exactly what that signal means and how directly it records changes in the ocean atmosphere system. This matters because the rise of large, complex organisms did not happen against a stable background. It happened while Earth systems were reorganizing. Oxygen is often placed at the center of the story, and for good reason. Animals require oxygen for aerobic metabolism, and larger or more active bodies generally impose greater metabolic demands. Yet the old narrative of a single late oxygenation event unlocking animal life has become harder to defend. Evidence suggests a patchy, dynamic ocean with oxygenated and poorly oxygenated environments existing across space and time. Some early animals may have tolerated lower oxygen levels than many modern animals. Local oxygenation could have mattered before global conditions became modern. There is another change that is easy to overlook because it does not produce a dramatic fossil by itself: the rise of algae and other eukaryotic primary producers. Biomarker and microfossil evidence suggests that eukaryotic algae became more ecologically important through the late Neoproterozoic. That shift may have altered the quality and distribution of food in marine ecosystems. More nutritious particles in the water and on the seafloor could support new feeding strategies. Again, there is no single switch. The ecological expansion of algae, oxygen changes, and animal diversification overlapped in complicated ways. The seafloor that emerged from this background was unlike most seafloors today. Before burrowing animals became abundant and deep bioturbation widespread, microbial mats often formed extensive, cohesive surfaces. These mats were not merely passive carpets. They trapped and bound sediment, affected local chemistry, and created feeding opportunities. They also changed preservation. A soft organism resting on or beneath a microbial mat had a better chance of leaving an impression than it might on loose, constantly reworked sediment. This helps explain the extraordinary Ediacaran fossil surfaces found in places such as Newfoundland, the White Sea region of Russia, Namibia, South Australia, and South China. A bedding plane can preserve dozens or hundreds of impressions at once, turning a moment of ancient seafloor into something like an ecological map. The map is incomplete. Soft tissues decay. Storms transport bodies. Sediment can deform. Different organisms preserve in different ways, but spatial patterns still matter. Paleontologists can ask which organisms lived together, how densely they were distributed, whether individuals avoided or clustered around one another, and whether growth forms changed with local conditions. One of the earliest famous communities comes from the Avalon assemblage, roughly beginning around 575 million years ago. Sites in Newfoundland and elsewhere preserve rangeomorphs and other frond-like organisms anchored to the seafloor. At first glance, they can look like plants, ferns, or sea pens. Those comparisons are visually tempting, but biologically dangerous. Plants had not colonized land, and the affinities of many rangeomorphs remain uncertain. Their branching architecture follows a fractal-like pattern, repeated units built from smaller repeated units. This allowed large surface area without the anatomy we associate with leaves, guts, or mouths. How did such organisms feed? Several hypotheses have been proposed. Osmotrophy, the direct absorption of dissolved organic compounds, was once widely discussed for rangeomorphs, though support varies among taxa and researchers. Suspension feeding or other interactions with dissolved and particulate nutrients may have been important for some forms. The honest answer is that we cannot watch them eat. Their bodies are preserved mainly as external impressions, and anatomy, such as digestive tracts, is usually absent. Any reconstruction should distinguish what the fossil shows from what a model proposes. The vertical structure of these communities is nevertheless important. Different frond-like species reach different heights above the bottom. That could reduce direct competition if they accessed resources from different parts of the water column. Some ecological studies of Ediacaran bedding planes suggest that these communities were structured rather than random. They were ecosystems, not simply collections of curiosities. The idea of an ecosystem is crucial because it moves us beyond the question, what was this organism? Even when identity is uncertain, ecology can sometimes be inferred from position, growth, association, and interaction. A holdfast indicates attachment. Repeated individuals of different sizes can reveal growth. Consistent orientation may record currents. Trails indicate movement. Scrape marks may indicate feeding. Borings in skeletons can suggest predation. Each clue gives us a verb, and verbs are often more informative than labels. By the Middle and later Ediacaran, the fossil record shows an expanding range of those verbs. Organisms attach, grow, absorb, filter, glide, graze, crawl, burrow, build skeletons, and perhaps attack one another. The transformation was gradual enough that no single fossil surface captures the whole process, but fast enough in geological terms to reshape the planet's biological identity within a few tens of millions of years. From our perspective, that is an almost impossible timescale to feel. 1 million years is already far beyond human intuition. The Ediacaran lasts close to 100 million years. Entire evolutionary radiations can rise and decline within it. When we compress the period into a documentary, organisms separated by 20 million years can appear side by side in our imagination. They were not. The Ediacaran was a sequence of changing worlds. The first of those worlds that truly announces itself in macroscopic fossils is quiet, rooted, and dominated by forms that seem architectural. The next becomes more diverse. Flattened bodies spread across matgrounds. Symmetries multiply. Movement becomes harder to ignore. And eventually the sediment itself begins to record the approach of a new ecological regime. On some Ediacaran seafloors, the most conspicuous organisms rose upward in dense communities. They resembled leaves, feathers, fronds, or underwater plants, which is why early interpretations sometimes placed them close to groups that now seem unlikely. Their appearance is one of the great traps of paleontology. Shape invites analogy, but analogy can disguise evolutionary distance. Charnia is the classic example. The first widely recognized specimen was discovered in Charnwood Forest in England in the 1950, in rocks then considered too old to contain macroscopic life. Charnia has a leaf-like outline divided into repeated branches. It was anchored by a discoidal holdfast, and its body extended upward from the seafloor. Similar rangeomorph organisms are now known from several regions. Their construction is difficult to compare directly with modern animals because the branching units repeat at multiple scales. The apparent simplicity of a frond hides a sophisticated growth problem. An organism attached to the bottom must acquire energy, maintain tissue, resist currents, and grow without being smothered by sediment. Some Ediacaran fronds reached substantial sizes. Their large surface areas may have been important to feeding or gas exchange. Population studies have also revealed size distributions, suggesting that some communities contained juveniles and adults, allowing researchers to think about recruitment and reproduction. Fractifusus, another famous rangeomorph from Newfoundland, did not stand upright in the same way. It lay along the seafloor as a spindle-shaped, quilted form with repeated branching units. Spatial analysis of Fractifusus populations has been interpreted as evidence for more than one reproductive mode, including the possibility of waterborne propagules combined with stolon-like clonal expansion. This is not the same as observing reproduction directly, but it shows how fossil maps can preserve biological information that a single specimen cannot. The Avalon communities also challenge a common intuition about early ecosystems, that the first macroscopic organisms must have lived only in bright, shallow water. Some important Ediacaran sites represent relatively deep water settings below the zone where photosynthesis would dominate. That is one reason the plant analogy fails. These organisms were not simply Precambrian seaweeds standing in sunlight. If the seafloor looked like a forest, it was a forest without wood, leaves, roots, insects, or vertebrates. More accurately, it was a vertical community of soft-bodied organisms occupying space above a microbial bottom. Their shapes influenced how water moved around them. Their holdfasts interacted with sediment. Their death and decay recycled nutrients. Even without modern predators, a dense field of bodies would create competition for space and resources. Ediacaran ecosystems were also not globally uniform. The familiar names Avalon, White Sea, and Nama refer to broad assemblage patterns recognized through time and geography, but they should not be imagined as three perfectly separate worldwide stages. Fossil occurrence depends on environment, preservation, sampling, and regional history. Still, the overall pattern is useful. Earlier communities tend to be dominated by rangeomorph and frond-like forms. Later communities include greater morphological and ecological diversity, including more mobile organisms and forms increasingly interpretable as animals. This changing diversity can be seen in body symmetry. Modern animals often exhibit bilateral symmetry, with a left and right side or radial symmetry organized around a center. Ediacaran fossils experiment with arrangements that are less familiar. Tribrachidium, for example, has three curved arms arranged around a central point. Its triradial symmetry is rare among living macroscopic animals. Computational work on fluid flow around its body has supported the idea that it could have fed by passive suspension feeding, directing water towards specific regions of the organism. Another form, Dickinsonia, consists of a flattened oval body with repeated units extending from a central axis. For decades, its identity was debated. Interpretations ranged across animals, fungi, giant protists, and extinct lineages. The discussion changed dramatically when organic molecules extracted from exceptionally preserved fossils were reported in 2018. The sterile pattern was interpreted as strong evidence that Dickinsonia was an animal. That result does not tell us exactly where Dickinsonia belongs within the animal tree, but it narrows the possibilities. The body itself remains strange. Dickinsonia could grow from very small individuals to specimens more than a meter long in some species. Fossil surfaces sometimes preserve repeated impressions, interpreted as evidence that individuals moved across microbial mats and fed at successive locations. Whether the animal digested the mat externally, absorbed nutrients through its underside, or used some other mechanism remains debated. What matters is that it was not necessarily a passive quilt lying where it grew. There is an important lesson here. Ediacaran organisms can look simple because their fossils are flat. Flat preservation strips away depth, color, texture, internal organs, and behavior. A modern jellyfish pressed into sediment would also become an ambiguous stain. A worm might leave only a groove. A soft sponge could be almost impossible to recognize without distinctive architecture. The absence of visible anatomy is not proof that anatomy was absent in life. At the same time, reconstruction has limits. It is easy for artists to give every Ediacaran organism eyes, mouths, tentacles, bright colors, and dramatic movements because those features make an image feel alive. Most would be inventions. For many taxa, we do not know color. We do not know whether they were translucent. We do not know how much relief they had above the sediment. We do not know whether some structures were internal or external. Good reconstruction should preserve uncertainty rather than hide it, so the safest visual language is restrained. Charnia can be shown as a soft, branching frond anchored upright, moving slightly in a current. Fractifusus can lie low on a matground. Tribrachidium can rest as a three-armed disc. Dickinsonia can be broad, low, and segmented-looking without giving it a face. The environment can do much of the cinematic work. Low-angle light in shallow water, suspended particles, rippled sediment at the edge of a microbial mat, distant fronds fading into haze. This world may appear peaceful, but peaceful does not mean static. By the time these forms were widespread, new types of organisms were beginning to leave evidence of directed movement and more active feeding. The seafloor was becoming something to travel across rather than simply attach to. And once bodies began to move with purpose, ecological relationships could become much more complicated. The temptation to sort every fossil into a modern category is almost irresistible. We find a flattened oval and ask whether it is a worm. We find a frond and ask whether it is a cnidarian. We find a disc and ask whether it is a jellyfish. These questions are reasonable, but the Ediacaran repeatedly warns us that resemblance is not ancestry. Some organisms may belong near the base of living animal groups. Others may represent stem lineages, branches closer to a modern group than to anything else living, but outside the crown group that contains all living members and their last common ancestor. Still, others may belong to extinct lineages that diverged before familiar body plans stabilized. The fossil record is therefore a mixture of relatives, experiments, and unknowns, rather than a clean parade toward the present. Consider Ernietta from the late Ediacaran of Namibia and elsewhere. It is reconstructed as a bag or cup-shaped organism partly buried in sediment, with a body made of repeated tubular units. Multiple individuals could cluster together. Modeling and sedimentological evidence have been used to explore how Ernietta may have interacted with currents and suspended food. It is visually distinct from a mobile bilaterian animal, yet it was a large organism occupying a structured community near the end of the Precambrian. Pteridinium is similarly difficult. Its body consisted of 3 veins meeting along a central axis, giving it a quilted, boat-like, or leaf-like geometry. Fossils may preserve it in different orientations, which complicates interpretation. It has been grouped with the so-called Ediacaran, but the broader affinities of these forms remain unresolved. They remind us that body architecture can disappear entirely from the later fossil record. Then there is Tribrachidium, whose threefold symmetry seems almost designed to frustrate comparison. It did not need to become anything familiar to be successful in its own environment. Fluid dynamics studies suggest that water passing over its surface could have been directed in ways compatible with suspension feeding. If so, an organism with a body plan, unlike any common modern animal, still occupied a recognizable ecological role. It extracted food from moving water. Ecological roles can therefore be more conservative than body plans. Filter feeding, grazing, attachment, and movement are problems that evolution can solve with different anatomies. The Ediacaran is full of cases where the function may be partly reconstructible even when the lineage is obscure. Dickinsonia sits near the center of this discussion because it combines strong evidence of animal affinity with an unfamiliar form. The 2018 biomarker study found cholesterol-related molecular fossils associated with Dickinsonia specimens, supporting an animal interpretation. But calling Dickinsonia an animal does not turn it into a primitive version of any single living animal. Its position within metazoa remains debated, and its mode of feeding is still reconstructed indirectly from fossils and traces. Its movement is especially fascinating. On some surfaces, paleontologists have identified repeated Dickinsonia-shaped impressions that have been interpreted as feeding traces. The animal appears to have occupied one patch, moved, then occupied another. The result resembles a sequence of ghostly body stamps. If that interpretation is correct, the animal interacted actively with the microbial mat, even though it lacked an obvious head in the fossil. Other fossils push closer to recognizable bilaterian behavior. Kimberella, known particularly from the White Sea region, had an oval body and is associated with fan-shaped scratch marks. These traces have often been interpreted as evidence that Kimberella grazed or scraped the microbial surface using a feeding structure. Its exact relationship to mollusks remains debated, and it should not simply be reconstructed as a modern slug. But it appears to have been a mobile animal with directional feeding behavior, a major ecological shift from a permanently attached frond. Directional movement changes the meaning of the seafloor. Once an animal can move toward food or away from unfavorable conditions, it can create spatial choices. A front and back become useful. Sensory information becomes valuable. Muscles and coordinated behavior become increasingly important. None of these traits necessarily appeared all at once, and some may have much deeper origins than the body fossils reveal. Yet trace fossils allow us to see when movement began to leave repeated, interpretable patterns in sediment. The smallest fossils can be as important as the spectacular ones. Echaria wariusha, described from South Australia, is known as a tiny body fossil associated in age and setting with horizontal trace fossils called helminthoidichnites. It has been interpreted as one of the oldest known bilaterians with a clear front and back, although details of how specific trace makers are linked to body fossils require caution. The organism was only millimeters long. In a documentary dominated by meter-scale Dickinsonia and tall fronds, Echaria would be almost invisible. Evolutionarily, however, a tiny directional crawler may represent a body organization with enormous consequences. Ylingia spiciformis from South China provides another striking piece of evidence. The fossil preserves a segmented, bilaterally symmetric animal, and one specimen is associated with a long trackway ending at the body. It demonstrates that complex, directed locomotion existed before the Cambrian. Again, this does not mean the Cambrian explosion was an illusion. The Cambrian still records a dramatic expansion in diversity, skeletonization, ecological intensity, and recognizable, animal clades. But some of the machinery was already operating. Even cnidarian-like animals may have appeared before the boundary. Aurora lumina attenboroughi, described from Charnwood Forest, has been interpreted as a crown group cnidarian from roughly 560 million years ago, although evolutionary placement of early soft-bodied fossils always invites continued testing. Its branching, tubular form is a reminder that familiar lineages did not necessarily begin with familiar-looking bodies. The same principle applies to sponges. Molecular estimates have long suggested Precambrian origins for sponges, but unequivocal body fossils have been difficult to establish. In 2024, researchers described Helicolos cantori from late Ediacaran rocks in South China and interpreted it as a crown group sponge related to glass sponges. Unlike modern glass sponges with mineralized spicules, it appears to have possessed an organic, lattice-like skeleton. The fossil, therefore, offers one explanation for why early sponges might be difficult to recognize if researchers expect mineralized support structures. These discoveries shift the question. Instead of asking whether animals suddenly appeared in the Cambrian, we can ask which animal traits, lineages, and ecological interactions were already present before it, and which became amplified after the boundary. The answer is uneven. Some Ediacaran communities still look profoundly alien. Others contain mobile bilaterians, cnidarian candidates, sponge-like forms, and skeletal organisms. The closer we move to the end of the period, the more the old boundary between strange Precambrian life and familiar animal life begins to blur. \n, the distinction between crown groups and stem groups is especially useful in deep time. A crown group contains all living members of a lineage, their last common ancestor, and every descendant of that ancestor. A stem lineage falls outside that crown, but is more closely related to it than to other living groups. Fossils on a stem can possess only part of the anatomy we associate with the modern group. This is why an early cnidarian need not look like a modern jellyfish, and an early sponge need not preserve the mineral spicules common in many living sponges. Evolution assembles characters over time. It does not deliver finished textbook diagrams at the first appearance of a lineage. \n, the same logic makes absence difficult to interpret. If a lineage began with tiny, soft-bodied representatives, its early record could be almost invisible. Molecular clock estimates can suggest that divergences occurred before unambiguous fossils appear, while fossils provide minimum ages for lineages we can recognize. Neither source of evidence is perfect. The strongest reconstructions come from comparing developmental biology, molecular data, trace fossils, body fossils, geochemistry, and precise geological dating rather than expecting one method to settle every question. Movement leaves a different kind of fossil record. A body fossil tells us that an organism existed. A trace fossil records something it did. A line across a matground can preserve direction. A branching burrow can preserve repeated decisions. A series of scratch marks can preserve feeding. A tunnel can show that an animal entered sediment and changed the environment beneath the surface. During much of the Ediacaran, the upper sediment remained relatively undisturbed compared with later Phanerozoic seafloors. Microbial mats could spread across broad areas. As bilaterian animals became more common and more active, this condition began to change. Crawling broke mat surfaces. Grazing removed microbial biomass. Burrowing mixed sediment and moved oxygenated water into new microenvironments. These effects are collectively part of a much longer transition, sometimes called the agronomic revolution or Cambrian substrate revolution. The term can make the transformation sound instantaneous, but it was not. Ediacaran trace fossils show that sediment disturbance began before the Cambrian and increased in complexity through the transition. Early horizontal traces are generally small and shallow. Later traces become more varied, and vertical burrowing becomes more significant. The important point is that animals did not simply evolve new bodies. They began engineering the physical environment. Microbial matgrounds were particularly vulnerable to this change. For an organism such as Dickinsonia, a stable mat may have been a feeding surface. For small grazers, it was a resource patch. For attached fronds, cohesive sediment could provide a particular style of substrate. As burrowing intensified, habitats based on undisturbed matgrounds became less widespread in many environments. Ecosystems changed not only because species competed or preyed on one another, but because animals altered the texture and chemistry of the seafloor itself. This is one reason the end of the Ediacaran cannot be described simply as a mysterious catastrophe wiping out a failed biota. Environmental stress may have caused major losses, and there is evidence for substantial turnover. But ecological replacement also matters. New animals could modify habitats in ways that made older lifestyles less competitive. A lineage does not need to be directly eaten to be displaced. It can lose the conditions on which it depends. Predation adds another layer. Evidence for predatory behavior before the Cambrian is limited but important. Cloudna, a small tubular organism that built a mineralized skeleton near the end of the Ediacaran, is among the best-known skeletal fossils of the period. Some Cloudna tubes preserve holes that have been interpreted as predatory borings. The holes show size selectivity and placement patterns consistent with biological attack in some studies, making them candidates for some of the earliest direct evidence of predator-prey interaction among animals. Cloudna itself did not resemble a shellfish in the modern sense. It formed nested, funnel-like calcified tubes and lived in marine settings that could include reef-like microbial and skeletal communities. Along with other late Ediacaran skeletal organisms such as Namacalathus, it signals a growing importance of biomineralization. Skeletons can support bodies, protect against damage, alter feeding, and change how organisms interact with sediment and water. Predation is one possible pressure favoring protective structures, but seawater chemistry and other ecological factors also influenced the evolution of mineralized skeletons. Namacalathus was once difficult to place, but is now often discussed in relation to Lophotrochozoan animals based on newer anatomical interpretations. It had a stalked, goblet-like form with openings in its cup. Its presence in carbonate environments, alongside Cloudna, contributes to the sense that terminal Ediacaran ecosystems were becoming structurally more complex. Hard parts were still rare compared with the Cambrian, but once mineralized skeletons appeared, they changed both ecology and preservation. The fossil record itself becomes biased toward these durable bodies. Meanwhile, mobile animals were becoming behaviorally more sophisticated. Trace fossils record not just movement, but changing patterns of movement. Recent work continues to examine whether Ediacaran trails preserve evidence of increasingly complex sensory behavior and resource tracking across the Ediacaran-Cambrian transition. Such interpretations are difficult because a trail is an indirect record. Yet a curved path, a repeated turn, or a response to a previous trail can reveal information about what an animal sensed and how it behaved. The emergence of sensory ecology may be one of the deepest changes of this interval. A sessile organism can survive by responding locally to current, chemistry, or light. A mobile animal faces a different problem. It must decide where to go. If food is patchy, movement must be efficient. If predators exist, detecting danger matters. If mates are separated, locating one another matters. Nervous systems, sensory cells, muscles, and directional bodies become parts of an expanding information economy. We should be careful not to project modern minds backward. There is no evidence of complex cognition in the human sense. The earliest bilaterians may have operated with very simple sensory networks, but even a tiny change in the ability to detect a chemical gradient or distinguish front from back can alter behavior. Once organisms move in response to information, ecology becomes increasingly interactive. This creates a feedback loop. Movement disturbs mats. Disturbed mats change resource patterns. Predation favors defense and detection. Burrowing changes oxygen penetration. Skeletons alter predator-prey dynamics. New feeding strategies change particle flow. Each innovation becomes part of the environment experienced by every other organism. The classic phrase Cambrian explosion emphasizes the rapid diversification that follows, but the fuse was already burning. By the final tens of millions of years of the Ediacaran, multiple ingredients of later marine ecosystems were present. Mobile bilaterians, suspension feeders, probable grazers, skeletal organisms, habitat engineers, and evidence consistent with predation. They were not yet combined everywhere with the intensity seen in the Cambrian. That is precisely why the Ediacaran is so revealing. It lets us watch ecological escalation before it becomes normal, and escalation came with losses. Some of the great quilted and frond-like forms disappear from the record. Diversity patterns shift. Geochemical evidence points to episodes of reduced oxygen availability. The final Ediacaran becomes a period of both innovation and instability, with old ecosystems under pressure while new ones spread. \n bioturbation also changes what later paleontologists can see. A stable microbial mat is excellent at preserving surface impressions under the right conditions. A heavily churned sediment destroys many delicate traces while creating other kinds of fossils. The rise of burrowing, therefore, changes both ancient ecology and the archive used to reconstruct that ecology. A decline in Ediacaran-style preservation can exaggerate the apparent speed with which soft-bodied communities disappear. \n this is one reason researchers compare multiple kinds of evidence across the boundary. Body fossils reveal anatomy, trace fossils reveal behavior, sedimentary structures reveal substrate conditions, and chemical proxies reveal aspects of oxygenation and nutrient cycling. No single record is continuous everywhere. When several independent records point toward increasing ecological disturbance and behavioral complexity, the case becomes stronger even if individual sites remain incomplete. Extinction stories are attractive because they offer clean endings. A community flourishes, catastrophe arrives, and the survivors inherit the world. The Ediacaran refuses to cooperate with that simplicity. Paleontologists recognize major changes in Ediacaran diversity, including a pronounced turnover between the White Sea and Nama assemblages before the final boundary. A 2022 analysis argued that decreased oxygen availability provides a plausible environmental driver for a major loss of Ediacaran taxa across this transition. The pattern is especially interesting because surviving organisms were more likely to possess traits associated with greater surface area relative to volume, which could help under oxygen stress. That result supports an environmental extinction mechanism, but it does not explain every disappearance across the entire period. Other researchers have emphasized ecological replacement. As mobile and burrowing animals expanded, they altered substrates and resource flows. The disappearance of classic Ediacaran forms may therefore reflect a combination of environmental stress, changing preservation, ecological competition, habitat modification, and true extinction. These mechanisms are not mutually exclusive. In a changing world, they can reinforce one another. Preservation complicates the picture further. Many Ediacaran organisms are known because microbial mats and unusual sedimentary conditions captured impressions of soft bodies. If those conditions became less common, organisms could disappear from the fossil record without disappearing immediately from life. This is sometimes discussed through the idea of a preservational Cheshire cat. The visible fossil record fades even if some lineages continue unseen. It is a useful warning, not a complete explanation. The terminal Ediacaran fossil record therefore matters enormously. Every site close to the Cambrian boundary can test whether supposedly vanished forms persisted, whether recognizable animals diversified earlier than expected, and whether ecological transition was abrupt or mosaic. South China has become especially important. The Dengying Formation spans part of the late Ediacaran and preserves multiple fossil assemblages. In 2024, Helicolos added a proposed crown group sponge to the picture. In 2025, the Tongshan Lagerstätte provided exceptional preservation from roughly the terminal Ediacaran, including rangeomorph fronds, and a diverse collection of organisms preserved in carbonaceous mudstones and shales. The preservation style gives researchers information that ordinary moldic impressions often lose. Then, in April 2026, researchers described the Jiangchuan Biota from Yunnan, a terminal Ediacaran assemblage preserved as carbonaceous films. More than a simple collection of strange Precambrian forms, the fauna includes organisms interpreted as showing connections with groups more familiar from the Cambrian. The authors described it as a transitional fauna. That phrase is important because it does not mean every Ediacaran organism was transforming into a Cambrian one. It means the community contains a combination of features that blur the traditional visual and evolutionary boundary. For decades, the Ediacaran-Cambrian transition was often presented as a contrast between two worlds: soft, enigmatic Ediacaran forms below, diverse, mobile, skeletonized animals above. The new fossil sites make the border more porous. Some recognizable animal groups have Ediacaran representatives. Some classic Ediacaran organisms persist late. Trace fossils show complex movement before the boundary. Skeletonization begins before the Cambrian. The Cambrian radiation remains real, but its roots reach backward. This is a familiar pattern in evolutionary history. Major radiations often look sudden because the fossil record crosses a threshold of abundance, preservation, and anatomical visibility. The genetic and ecological groundwork may begin earlier. Once skeletons become common, fossils become easier to find. Once organisms grow larger or enter new environments, they leave more traces. Once ecosystems intensify, the rate at which bodies and behaviors generate fossil evidence can increase. The end of the Ediacaran also reminds us that periods are human divisions imposed on continuous time. The formal boundary is defined geologically, not because every ocean changed on the same morning. Organisms did not know they were leaving the Precambrian. Local ecosystems responded to local conditions, while global evolutionary trends unfolded over millions of years. Imagine standing on a seafloor near that boundary. In one region, microbial mats may still dominate and soft-bodied forms remain common. In another, burrowers are mixing sediment. Elsewhere, small skeletal tubes rise from carbonate surfaces. Tiny predators or grazers may be interacting with prey. A sponge-like animal filters water through an organic framework. A bilaterian leaves a track. The future is present, but unevenly distributed. This mosaic helps explain why arguments about the Cambrian explosion can become confused. If explosion means the instantaneous origin of animals, the fossil record contradicts it. Animals existed earlier. If explosion means a geologically rapid diversification of many animal body plans, ecological roles, skeletal forms, and abundant trace fossils during the early Cambrian, then it remains one of the central events in the history of life. Both statements can be true. The Ediacaran contribution is to show that revolutions have prehistories. Before the Cambrian seas filled with trilobites and complex food webs, there were already experiments in movement, feeding, symmetry, skeletonization, and habitat engineering. Some experiments disappeared. Some may have survived in altered form. Some ecological strategies became foundations of the Phanerozoic world. What vanished, then, was not simply a list of bizarre organisms. A style of ecosystem vanished. Matgrounds became less dominant in many settings. Large, quilted organisms no longer defined seafloor communities. Rangeomorph forests disappeared. The balance between sessile and mobile life changed. Sediment became more thoroughly worked. Interactions became more intense. Extinction is therefore only part of the ending. The deeper story is replacement of one ecological world by another. \n\n, there is also a geographic caution. A turnover seen in one basin may not occur at exactly the same time or in the same way in another. Sea level, water depth, local oxygenation, sediment type, and preservation all influence which organisms are recorded. Global narratives are built by correlating many local sections, each with its own gaps. That makes precise radiometric dates and chemostratigraphic markers especially valuable near the Ediacaran Cambrian boundary. /n the newest Lagerstätten are powerful partly because they fill ecological and preservational gaps rather than merely adding new species names. Carbonaceous films can reveal outlines and structures that molds fail to retain. Fine-grained deposits can preserve small animals that would disappear in coarser sediments. A site that samples a different water depth or substrate can expose an entire community absent from better-known localities. The history of early animals changes when the sampling window changes. At approximately 539 million years ago, the Ediacaran gives way to the Cambrian. Geological time crosses a formal boundary, but evolution does not reset. There is no biological curtain falling between acts. Early Cambrian rocks record a striking increase in trace fossil diversity and complexity, the spread of biomineralized skeletons, and the appearance or expansion of many groups that can be linked more clearly to living animal phyla. Within the following tens of millions of years, ecosystems include arthropods, priapulid-like worms, early chordates, brachiopods, sponges, cnidarians, echinoderm relatives, molluscan lineages, and many forms that remain difficult to classify. Famous sites such as Chengjiang and the Burgess Shale preserve the later consequences of this diversification in extraordinary detail. Why did the Cambrian radiation happen? There is no single accepted cause. Hypotheses involve oxygen availability, developmental genetics, ecological interactions, predation, changes in nutrient cycling, seawater chemistry, skeletonization, sensory innovation, and environmental restructuring. These mechanisms can interact. A new predator changes selection on prey. A new skeleton changes both defense and fossilization. A burrower changes sediment chemistry. Greater oxygen availability can support more active behavior, while active ecosystems can themselves alter carbon and nutrient cycles. The Ediacaran makes clear that many ingredients predate the formal boundary. Bilateral movement appears before it. Grazing appears before it. Skeletal tubes appear before it. Probable cnidarians and sponges may appear before it. Complex ecological communities certainly appear before it. The Cambrian did not invent complexity from nothing, yet continuity should not be exaggerated either. The scale and abundance of Cambrian ecological innovation are remarkable. Trace fossils penetrate sediment more deeply and diversify. Hard parts become common in many lineages. Predation intensifies. Eyes and other sensory structures become prominent in the fossil record. Food webs develop new vertical and behavioral dimensions. The seafloor itself becomes increasingly churned. This is why the idea of an information revolution has become an interesting way to think about the transition. As organisms sense more, move more, and interact more directly, survival depends increasingly on processing environmental information. A predator must find prey. Prey must detect danger. Grazers must locate food patches. Burrowers must navigate chemical gradients. Even without complex brains, selection can favor better sensors, faster responses, and more coordinated movement. The physical record of those changes may be humble. A tiny winding trail in ancient sediment can represent a major behavioral shift. The trackmaker does not need to be large or visually dramatic. It needs only to make choices that matter. From this perspective, the quilted giants of the Ediacaran occupy a poignant place. Some were large and successful for millions of years, yet the ecological future belonged increasingly to organisms that could move, burrow, bite, filter, scrape, sense, and build. Size alone did not determine the winners, but the word winners is also misleading. Evolution has no final tournament. Many Cambrian lineages later disappeared. Many modern groups would suffer mass extinctions and recover. The history of life is not a ladder from primitive to advanced. It is a branching history shaped by contingency. The Ediacaran demonstrates that vividly because it preserves branches that seem to lead nowhere familiar. Their disappearance makes them feel experimental, but every lineage is an experiment in the same retrospective sense. We call some successful only because descendants happen to survive into our present. There is another reason to resist treating the Ediacaran as alien failure. Some of its organisms were animals, and some animal lineages clearly persisted. Sponges, cnidarian relatives, and bilaterians have roots extending into the Precambrian. The boundary between alien and familiar is partly a limitation of our own pattern recognition. A Dickinsonia does not look like an animal because our intuitive animal is mobile, three-dimensional, and equipped with a mouth, muscles, and sense organs. But animal evolution did not begin with our intuition. It began with cells cooperating in bodies that solved problems in ways we are still reconstructing. A sponge does not move across the seafloor, yet it is an animal. A placozoan has no neurons or muscles in the conventional sense, yet it is an animal. Modern biology itself warns us against defining animal life by a narrow visual stereotype. The Ediacaran expands that warning into deep time. The new Chinese fossil assemblages sharpen the point. Tongshan preserves classic Ediacaran-style forms close to the end of the period. Jiangchuan preserves a community interpreted as bridging anatomical expectations across the transition. These sites suggest that the final Precambrian oceans contained more evolutionary overlap than a simple before and after diagram can show. They also remind us how much can change with a single new deposit. Paleontology is constrained by where rocks of the right age are exposed, where soft tissues were preserved, where researchers can collect, and which specimens happen to survive erosion and tectonics. A major gap in the fossil record may reflect biology, geology, or both. Every newly discovered Lagerstätte has the potential to reshape the sequence. That means the story we tell today is provisional. A future fossil could push a lineage farther back. A new chemical technique could challenge an old classification. Better imaging could reveal anatomy hidden in a carbon film. More precise dates could rearrange a sequence. The Ediacaran is not a solved puzzle with a few pieces missing. It is a research frontier disguised as ancient stone. And that may be the most important difference between how this period was once presented and how it can be presented now. We are no longer looking only at mysterious fossils before the Cambrian. We are looking at ecosystems in the act of becoming more animal-dominated, with multiple lineages and behaviors appearing at different times, under changing environmental conditions. More than 500 million years separates us from the last Ediacaran seafloors, but their legacy is not abstract. Every modern animal ecosystem depends on innovations that were being assembled then or earlier. Multicellular bodies, cell signaling, adhesion, development, feeding, movement, sensory response, reproduction, and ecological interaction. We cannot point to one Ediacaran organism and call it the ancestor of all later animals. Evolutionary relationships are rarely that direct in the fossil record. What we can see is a landscape of possibilities. Some organisms grew as branching fractal forms, some spread as quilted sheets, some adopted radial or triradial symmetry, some crawled, some scraped, some burrowed, some built skeletons, some filtered water. The diversity of solutions appeared before the modern caste was fully established. The Ediacaran also changed the planet physically. Animals are geological agents. Burrowing mixes sediment. Grazing changes microbial communities. Skeletons create new carbonate structures. Filter feeders move particles through water. Predators change population structure and selection. Over time, biology modifies the environment in which future biology evolves. That feedback did not begin in the Cambrian. The Ediacaran records its early stages. A microbial mat once covered the seafloor where a mobile animal later crossed. The trail broke the surface. Another organism encountered the disturbed patch. Sediment chemistry changed locally. A tiny behavior became part of Earth history. The scale is almost paradoxical. The transformations were enormous, but many of the actors were small, soft, and easily erased. The fact that we know them at all depends on unusual preservation. Volcanic ash, storm deposits, microbial mats, carbonaceous films, mineral coatings, and the chance exposure of ancient bedding planes hundreds of millions of years later. A fossil surface in Newfoundland or South Australia is therefore more than a collection of specimens. It is the surviving geometry of an ecosystem. Distances between organisms record space. Body sizes record growth. Orientation records current or attachment. Overlapping traces record sequence. Chemistry can preserve molecules from vanished tissues. The stone is not alive, but it retains the consequences of life. This is why the newest discoveries matter so much. The 2025 Tongshan Lagerstätte and 2026 Jiangchuan Biota add new detail at the exact part of the timeline where older narratives were most tempted to draw a sharp line. They show that the terminal Ediacaran still held classic forms while also revealing organisms that make the coming Cambrian world less abrupt. The result is not a weaker story. It is a better one. A sudden miracle is dramatic, but a long ecological transformation is more revealing. It shows evolution working through accumulation, interaction, and contingency. It allows us to see that major transitions are built from many smaller transitions that overlap without synchronizing. Some Ediacaran lineages were probably lost completely. We may never know what living organism would have looked most like a rangeomorph, because there may be no living equivalent. Their disappearance narrows the range of body architectures available for comparison. Modern biodiversity is vast, but it is still only the surviving fraction of life's historical possibilities. That idea changes how we look at an Ediacaran fossil. A strange three-armed disc is not interesting because it failed to become modern. It is interesting because, for millions of years, three-armed symmetry was a workable solution. A quilted body is not primitive because it lacks a skeleton. It belongs to an ecosystem in which skeletons were not yet the dominant currency of survival. A rooted frond is not a plant mimic. It is its own answer to the problem of living above a microbial seafloor. Near the end of the period, those answers met new pressures. Oxygen conditions shifted. Habitats changed. Mobile animals disturbed mats. Skeletons spread. Predation likely intensified. More lineages entered ecological space. The result was not one clean replacement event, but by the early Cambrian, the average marine ecosystem was moving toward a structure that would be more recognizable to us. Recognizable does not mean modern. Cambrian animals could be just as strange as anything in the Ediacaran, but the underlying grammar of the ecosystem becomes familiar. Predators pursuing prey, animals burrowing through sediment, armored bodies, active swimmers, complex sensory organs, and densely connected food webs. The stage has changed. What survives from the Ediacaran is therefore both ancestry and absence. Some roots of modern animal life extend into it. At the same time, entire styles of body construction and community organization vanish. The period is a bridge built partly from lineages that crossed it and partly from lineages that did not. There is humility in that record. We often describe deep time through the organisms that lead toward us because those are the branches we recognize. But most evolutionary branches do not lead to the present. Their importance is not reduced by extinction. They shaped environments, interacted with neighbors, and occupied Earth for spans of time vastly longer than recorded human history. If we could return to an Ediacaran sea, we would probably misidentify almost everything at first glance. We might mistake animals for plants, colonies for individuals, feeding surfaces for simple textures, and traces of movement for random marks. We would search for faces that did not exist and overlook organisms only millimeters long that carried the architecture of the future. The longer we watched, the more the scene would change. A Dickinsonia would no longer be a static imprint, but a living body interacting with the mat. A tiny bilaterian would cross a surface with direction. A frond would bend in a current. A tubular skeleton would rise from carbonate substrate. Water would pass through a sponge-like lattice. Life would become less alien, not because the organisms changed, but because our expectations did. And eventually, we would understand the central lesson of the Ediacaran. The age of animals did not begin when animals became easy for us to recognize. It began earlier, quietly, among soft bodies and microbial seafloors, while the rules of complex ecosystems were still being written. The Cambrian did not erase that world. It inherited it, transformed it, and buried most of its shapes beneath the success of new ones. The fossils that remain are fragments of a time when animal life had already begun, but the future had not yet decided what an animal world would look like. A final way to measure the distance between that world and ours is to think about what is missing from the soundscape. There are no waves breaking against forests of land plants because land is bare. There are no fish splashing at the surface. There are no shells rattling in the surf in the abundance familiar from later seas. Most of the drama takes place slowly, at the scale of currents, films of microbes, millimeter trails, and bodies pressing into sediment. Yet this quiet world contains one of the loudest changes in the history of life when viewed across geological time. Biology is becoming capable of reshaping marine habitats at a new scale. The seafloor is beginning to move because animals are moving through it. Food webs are becoming more interactive. Bodies are becoming more varied in how they acquire food, defend themselves, and respond to surroundings. The Ediacaran is therefore best understood neither as an alien dead end nor as a simple prologue. It is a complete chapter of Earth history with its own dominant forms, its own ecological experiments, and its own extinctions. It is also the chapter in which the foundations of later animal ecosystems become increasingly visible. Every new fossil site changes the balance between those two truths. The stranger the fossils appear, the more we appreciate the lost diversity of early complex life. The more recognizable features we discover, the deeper we push the roots of the animal world. Both processes are happening at once. For now, the stones keep the remaining evidence. A branching imprint, a carbon film, a trail across a mat, a tiny tube with a hole through its wall. None looks dramatic by itself. Together, they describe a planet learning a new biological language.